Well, so before, I would like to welcome everyone to the Theoretical Computational Biophysics seminar. Today we have Professor Dr. Frank Noe from Free University Berlin as a speaker. Before I introduce Frank, I would like to remind everyone to please turn off your microphones. And if you wish, you are encouraged to turn on your camera so we can see your face, but it's completely up to you. And then for questions and answers, if there are really pressing questions, I hope Frank doesn't mind to be interrupted maybe once during the talk or maximum twice, but it's best if you actually keep the questions for last. So when we are done with the talk, you can type in your questions in the chat box or just raise your hand indicating that you have a question. I'm going to call on you, asking you to actually ask your question after turning on the microphone. So that way you might have better interaction with the speaker. So with that, it's really my great pleasure to introduce Professor Frank Noe from Freie Universität Berlin. So Frank received his bachelor's degree in electrical engineering from Stuttgart, Germany. And then following that, he received the Master's of Science in Computing from Ireland. And then he moved on and received his PhD in computer science from University of Heidelberg and the Interdisciplinary Center for Scientific Computing, the summa cum laude, actually a degree from Heidelberg. So you can see he developed really a strong foundation in engineering, computing, and computer science. And then he moved on into his postdoc in Heidelberg, again, staying there where he started applying modeling and simulation techniques with this strong foundation to biological systems. Since 2017, he has been with Free University in Berlin, initially as a research leader, as a group leader. But since 2013, he has been a professor in mathematical modeling in life sciences in the university. And since 2016 or 15, he has also co-appointments at the chemistry department at Rice University. Frank has been doing really excellent, exciting, outstanding work in terms of developing modeling and algorithms for biophysical and biological systems in general. And as you can tell from his very diverse and complementary background, so he has been very successful in combining computer science techniques like machine learning, neural networks, deep neural networks, combining them with the StatMake problem like molecular simulations that many of us are interested in. So he has been very well regarded in the field with very high impact papers. I was just checking his page and noticing that several of his papers have received several hundreds of citations. Just last year, Frank received almost 3,000 citations to his high impact work. So we are very excited to have you here, Frank. Thank you again for accepting our invitation and being with us, and we look forward to your seminar. It's all yours.
Thank you very much, Emad, and everyone for having me and organizing this very nice lecture series. So yeah, let me share my screen. So hopefully you should be able to see the slides and my cursor. Yeah, okay, great. So I'll start then. So yeah, I guess most of you probably know me and my group for our work on Markov state models, but I will be talking mostly about deep learning for molecular physics problems, and that includes biophysics, but it's not exclusively biophysics. And some of the things I'm going to present today are like more recent work and perhaps more, things that could be like door openers in certain problem areas and are maybe not ready to use on, not all ready to use on very large scale problems, but I think it's still important to go in these directions. And so in the last couple of years, we've been very excited about combining machine learning, artificial intelligence with molecular science problems. And so clearly, you have seen many examples of AI and machine learning in various real-world applications, such as in robotics or reinforcement learning for playing games, such as DeepMind's Go. And in all of these applications of machine learning and AI, we have to essentially solve two problems. We have to somehow frame our problem as an optimization problem, because eventually all machine learning approaches at the moment are performing some sort of optimization problem. And secondly, we have to define what is our learning structure. So what is the algorithmic structure or the data structure that holds the parameters that we want to optimize? And is there anything particular about the structure? And now when we are in physics, in molecular physics or other sorts of physics, we have usually a strong foundation. We have things like certain physical laws, we have equations, and we have certain principles that we want to obey with the solutions of our physics problem. And so often physics tells us what we want to do in terms of the optimization problem. So often physics defines what is the optimization problem. And often physics has a lot to say about what is the learning structure. So for example, are there certain constraints or is like there a certain manifold in which the solution should live? Are there certain symmetries, like for example, the energy of a molecule in vacuum or implicit solvent should be invariant to translation and rotation, and if it's a quantum model, it should also be invariant to exchange of identical particles, etc. So things like that. Physics tells us a lot about how the architecture of our machine learning model should look like, and it's a good idea to incorporate this knowledge so that we don't have to learn that, and that can help us quite a lot in getting better solutions with the help of machine learning. Now, we are quite interested in applying these methods on making some progress on fundamental problems, so such as the sampling problem, statistical mechanics, where you basically have an energy surface defined in some way that could be an empirical potential, and then together with thermodynamic variables, this defines a probability distribution that you want to sample from. But we know sampling is hard, especially for large molecules, because we have many probability modes or metastable states in this high-dimensional energy surface, and there are rare events connecting them. So it's a challenge sampling from the surface, even though we know what a probability density is. So we can easily probe the energy of the probability density without a normalization constant at a certain point, but it's not easy to sample from it. Okay, so that's one problem. And another problem that I will be talking about at the end is quantum mechanics, the electronic structure problem. So how to efficiently compute, for example, ground state energies and ground state wave functions for molecular problems. Okay, so let's start with the sampling problem using an example. This is Barney's bar star, so two small proteins, and we're interested in the association and dissociation of these two proteins. If we simulate them in explicit solvent, we need about 100,000 atoms for essentially the smallest box that we should at least build to simulate this process. And so these numbers are not completely new anymore, but roughly we need I'm trying to my thumbnails here so that they can see my slides. All right. Roughly, we can do something like a microsecond in a week, probably in a few days now with the newest GPUs, and but the mean time to bind is about 50 microsecond in this box size. So that means one GPU year, and the mean time to unbind is greater than 1 hour for the wild type of Barney's bar star, or 1 million GPU years. So obviously it's not a good idea to just run simulations and hope for the best. What can we do about that? There are, of course, many approaches to enhance sampling somehow. one approach that can help us quite a bit are Markov state models, because it allows us to combine simulations that that may be individually short into statistics of the long-term dynamics. So we don't necessarily need to see all the rare events in single trajectories, which makes the sampling problem a lot easier. And a lot of research has been done by our group and other groups on how to best build Markov state models. And yeah, I want to come to a way to build these with deep learning, because deep learning can help us do the job of building a Markov state model well. And for that, we need to understand what is the optimization problem that we want to solve, and what's the kind of machine learning structures that we want to have, as I said in the beginning. So what's the optimization problem that we want to solve when we want to build a good Markov state model? So we can learn that by looking at the mathematics underlying what's happening in molecular dynamics and what's happening if we build an MSM. And a useful view there is that of the dynamical propagator. So basically the idea is we are not looking at a single trajectory and how it evolves in time, but we look at the statistics of many, many molecules over time. So an ensemble of molecules. And we start with an initial distribution rho 0, and we propagate that for some time tau by just running essentially molecular dynamic simulations on molecules sampled from the starting distribution for some time, and then our probability distribution propagates. Right, so this is just a hypothetical idea so far, right? So, but you can imagine doing this by just starting many simulations from some initial distribution. Could be a delta distribution, could be all the same structure, and just running them in parallel. So that's the idea here. So mathematically, we can write what's happening here under the dynamics as a spectral decomposition of this operator. So basically, that means if we run in equilibrium, what basically happens is that we can represent the probability distribution at any time by a linear combination of these eigenfunctions. So that's basically basis functions that can be used to represent our probability density at any time. And so these are like modes, and these modes decay exponentially in time until there's only one mode left, and that's the equilibrium distribution. So that's when you have a number of molecules in every state that you find in equilibrium. Okay, and so here's the picture for this in a simple one-dimensional energy landscape with 4 metastable states. So here you would have 4 eigenvalues that are close to 1. These are the eigenvalues, e to the minus lag time times rate of decay of that process. So that means there are 4 slow processes because there are 4 long-lived states. And we can associate them to certain events on the transition events on the energy landscape. So this here, the red guy, that's just the equilibrium process. So that's not so interesting. Then here, the yellow one is the transition between the AB and the CD state. So the transition across the highest energy barrier, and that has the highest time scale associated with it. And then here, the next lower time scale, that is the transition between A and B. And then here we have the transition between C and D. And you see this if you look basically at where the sign changes in the eigenfunction. So that basically tells you where the transition is. So in other words, these eigenfunctions are telling you how this high dimensional dynamics process can be decomposed into transitions between long-lived sets. So they can be used in order to decompose your high dimensional energy landscape into sets or clusters of states that are long-lived and between which the rare events are happening. Okay, and so the aim now, the aim of our learning problem to try to build an optimal MSM is to approximate these eigenvalues and these eigenfunctions as good as possible. Now, how do we do that? Because these are theoretical quantities. Yeah? We know they exist, but we don't have them in any practically tractable form. Yeah? You would these are things that live in a high-dimensional state space, and so you need to sample infinitely many MD trajectories to approximate them, to really compute them exactly. So how do we do that? It turns out that we can write this approximation problem, find the best approximation to the eigenvalues and the eigenfunctions of the molecular dynamics as a variational approach. And so the theorem says basically look for functions. So look for candidate functions or ansatz functions such that the time autocorrelation between them is maximal. If you sum up the time autocorrelations of over all these functions. Additionally, we have the constraints that they need to be normalized, so we cannot just maximize trivially by ramping up the amplitudes. And additionally, we also require that these functions are orthogonal to each other, so that we're not finding the same process over and over again. And then if we have done this, if we have maximized this sum, we have found the exact eigenfunctions, and the sum of this maximization problem is actually the sum of eigenvalues. So, but this is something we can actually compute, because we can just try some functions. We can just kind of make up some collective variables, project our dynamics on them, by using molecular dynamics simulations, just project them on these collective coordinates and compute autocorrelation functions. And now all we need is some sort of mechanism to try out many functions and maximize this score, this variational score, right? There's a generalization to this. So this variation approach for conformation dynamics is for equilibrium dynamics. There's also an approximation, a generalization that's valid for non-equilibrium dynamics. So if you have, let's say, an electric field across a membrane, or you are pulling on a molecule or something like that, you have something time dependent, then instead of maximizing eigenvalues, you basically want to maximize singular values. But in the case that you simulate at equilibrium conditions, those are the same things. So now, how can we optimize these variational scores? So one option is to do this linearly. You just define some basis set of functions, and you maximize the coefficients to linearly combine them so as to satisfy this principle. And then what you get is the TICA method, the time-lag independent component analysis method. And also what you get is, what you can get is a Markov state model. Namely, then if your functions, if your basis set are just characteristic functions, so if they're just essentially cluster membership functions. But we can also use a neural network to do this. And if we use neural networks and then use the VAMP score to optimize for finding the best representation of the slow processes, then that's called a VAMPnet. And the way to use it is as follows. We take trajectories, so simulation data. We take it at time t and t plus tau. So basically we look at kind of lagged simulation frames in our data, just push them through the network. And the network does basically all the things that we used to do sort of manually in a Markov state model construction pipeline. So we start at a high dimensional state space or feature space. We reduce the size of this feature space, and we eventually arrive at a representation where we could, for example, just have a layer which is called softmax layer. So that's basically a clustering layer. So every neuron at the output could just represent a membership or probability that our configuration belongs to a certain Markov state. Yeah, so these could be just values between 0 and 1, and they sum up to 1. Yeah, so then this is basically doing a clustering. And then we can directly get the Markov state model by computing simply correlation matrices on the output and transforming them to a transition metric. So this directly gives us a Markov state model. And it tries to learn essentially all the dimension reduction, transition matrix estimation, coarse graining of the transition matrix, TK, all of this stuff end to end within the neural network. So this can really help us to kind of get better models and avoids maybe poor decisions that we would otherwise be in danger to make on the way. And so here's one example where we use this. This is serine protease, TMPRSS2. That's important for SARS-CoV-2. It's one of the proteins, the receptor proteins on host cells that are used by the virus to enter, for example, into lung cells. And here we are studying the molecular mechanism of drug molecules, and we use these models to try to develop better drug molecules with our collaborators at the NIH and in Göttingen. And the starting point was that we didn't have a structure, so still don't have an experimental structure for TMPRSS2. We have homology models, of course, with all their problems, but a homology model may be a good guess for a serine protease, because we know a lot about the structure of serine proteases. So we started from a homology model. We did hundreds of microseconds of simulations, adaptive simulations using VAMP nets in order to get models on the way and make decisions of where we want to simulate next. And then, yeah, so here's just an overview of what this protein is doing. So it's basically interacting with the S protein of the SARS-CoV-2 virus before that can bind to the ACE2 receptor and then enter the cell. So this is an important step for the virus. And if you inhibit that, the virus can probably not enter anymore. And so here we are looking at drugs, Carmostat and Afamostat and others, and also try to develop new ones with our experimental collaborators that do in vitro assays, but also cellular assays where they look at really the cell entry of the virus and whether that can be inhibited by adding certain drug molecules. And so what we get out of our Markov state models is, first of all, an explanation why these two molecules, Carmostat and Afamostat, that are actually drug molecules that are approved on the Japanese market. Why they, first of all, they inhibit quite well, and why they inhibit differently. And that's kind of surprising because both molecules are covalent inhibitors. So we're not simulating here the covalent state here, which would be on the right. But so there's essentially a part of the drug molecule which gets cleaved by the enzyme, so by the serine protease mechanism of the enzyme, and fused. That part is fused to the enzyme and thereby blocks the binding pocket for the spike protein. And this covalent group is the same for both drug compounds. So if the covalent inhibition mechanism is the whole story, they should be doing exactly the same. They should have the same K on, K off, and binding affinity, but they don't. Now Afamostat inhibits a lot better than Carmostat, and the reason I think why this is, that's something you can understand when you look at the Markov state model of the state before you enter the covalent inhibition state. And so you see here that the kind of the reactive intermediate state has quite different populations for these three compounds here. So Carmostat and Afamostat is what I mentioned before. GBPA is just the metabolic form of carmostat that's relevant in the human body. So in the blood, there will be a reaction that turns carmostat very rapidly into GPPA. So really, that is the form we should be interested in. And so you see here that the lifetime of the nafamostat reactive state that could react to this covalently inhibiting state is much more probable. And so, and the reason for that is that basically the stability of these intermediates just before the covalent inhibitory state, that they depend on the leaving group, right? They depend on what differs between these different drug molecules. Yeah, so this is an example where Markov state models can help us to, first of all, sample the relevant structures of the protein that we cannot easily access at the moment with the experimental data. Even from experimental data, if we had a structure, we would still only have one structure. And secondly, try to understand the mechanism and help to explain how different drug molecules differ. So, and currently we're using these insights and also these protein ensembles to try to optimize these drugs and to find new drugs. Okay, so finishing this part, Markov state models learned with, for example, deep learning methods can help us to sample more efficiently because they help us to break down the sampling problem into small manageable pieces. And we can decide which pieces we want to simulate by using reinforcement learning or active learning on the Markov state modeling. So we can iteratively update our belief what is the best choice to start new simulation trajectories from in the next round by improving on the states where we have little statistics so far. And therefore we can improve our statistics. And for example, going back to this Barney's bar star problem, we can completely sample association and dissociation and intermediates for this problem in a total amount of 2 millisecond simulation data, which is a lot less than the hours or maybe days of data we would need to collect in equilibrium. And then we can get actually a Markov state model of protein-protein association and dissociation. It's still very expensive to do that. So in this particular case, you need about 20,000 GPU days to get these 2 millisecond molecular dynamics. Maybe now we need a little less with modern GPUs. And if you compare that with the combustion energy of Saturn V rocket or the energy it takes to transport a 15-ton payload to lunar orbit, that's 1500 gigajoules. So we are burning a lot of rockets here. But that's what computational scientists do. But hopefully we can maybe improve on these methods and make them more efficient so that we get more out of the rockets we're burning. There was a question, happy to take that now before I go to the next part.
Avi, you had a question?
Yes, I do. Frank, thank you. So the question that I had was, when you learn, or when any algorithm learns literally, the algorithm is learning the mean as well as learning the noise. So especially when one is looking into extended sampling, the means are learned quite well, but one has to be arty about learning the noise. Is there any sort of decisions that needs to be made when this noise is being learned? So an extensive set can be understood. So that was my question.
So in these examples, Markov state models, also the next part, we are learning something about the probability distribution of a process. So there's not really a thing like noise in that sense. I mean, of course, there's always noise on all of the parameters that we are estimating. Mm-hmm. Yeah, and we can with ways to understand what the effect of that noise is. So for example, when we build a Markov state model, what we can do is we can do bootstrapping. Mm-hmm. cross-validation. So we can train on parts of the data and test on other parts of the data. And we can test if the qualitative statements that we make. So for example, when I made the statement, nafamostat is a better drug than camostat because this intermediate state is more populated. Now we can test the statistical significance of this statement. And these little gray numbers here are actually the uncertainties that we get out of bootstrapping. And then we can test if this statement holds under statistical significance. And that's an important thing to do.
And like, if I may just continue a bit on that. So, so essentially of the several microseconds of simulation that was done before, there is a finite possibility of actually finding rare structures which are still correct and that are not there within the original pool of structures.
Sure.
So would you comment on that? Like, how would that be feasible if essentially that I would somehow, the autoencoder would have to, by using some level of sigma, so to speak, some noise, find certain regions that were not there. And that would be a new, let's say, an add-on to the several microseconds of investment that have already been made. So would there be, like, why would that happen? Like if the noise is not, like when I say noise, I mean like the low probability region of the density. So, so what would, is there a straightforward way of thinking about it? So yeah, we can answer this later, but that was my thought process.
Yeah, so perhaps you can go into more detail later. So there's no autoencoder here, right? This is just essentially learning a low-dimensional representation of the data from which you can build Markov state models efficiently. And so this is essentially just a representation of the data in a more convenient form, if you like. So the method does not find, this method, the machine learning method doesn't find anything that's not in the simulation data.
I see.
And what helps is this active learning where you decide based on what you have simulated, and perhaps you have found a few states that you have very poor statistics on, and then you can decide to simulate more in these states and improve your data. And to make this decision efficiently, it's useful to have a simple representation of your data.
I see, understand that. Now I understand. Okay, got you. So I think it's a way of identifying.
But it's always possible that there are some states that you haven't seen, and then you can't say anything about them with any simulation method. I think there's no magic pill out of that. Yeah.
Sure. Okay. Thanks. Yeah.
Sure. So about this burning of rockets. So here is an idea that I think may be useful to leverage some of the modern machine learning tools that can help us with the sampling problem in statistical mechanics. And this is very early stage. But I think it's an important idea because essentially all sampling methods and also all enhanced sampling methods that are established are built upon either molecular dynamics or Markov chain Monte Carlo. So they're all built on algorithms that do little steps in state space and then accept or reject them, or make sure that they somehow sampled from equilibrium in the very long run. And then maybe you have some algorithm on top of that, for example, generalizes across ensembles like replica exchange, or you have things like metadynamics or umbrella sampling where you add bias potentials, etc. But it's always built on an algorithm which does small steps, because we know it's hard to make large steps in a complicated condensed matter system like a solvated protein. Yeah. But what if we had a tool that helped us to learn to make large steps? And then maybe we could do things like people do routinely in the Monte Carlo simulation of simple solid models like Ising models, where you define large-scale moves. And so here, this line of research, Boltzmann Generators, is essentially trying to make some progress in that direction. So can we somehow hope to make more direct samples of the probability distribution defined by our energy function and our thermodynamic constraints, such as temperature and pressure and number of particles? And there's a very interesting line of research in deep learning, which is called generative deep learning. And the idea is as follows. So let's say we have images from a database of celebrities, for example, and we want to learn a probability distribution over these images so that we can sample new images, and they look like celebrities, but they're like new celebrities. So like made up people that are not in the database. You know? Basically the field of directed generative networks does this as follows. So we have a probability distribution at the input of a neural network that is easy to sample, such as a multidimensional Gaussian distribution. And then we learn a machine learning model, such as a neural network, which transforms these simple random variables into samples of the complicated probability distribution that we're interested in. And then, of course, we have to define how to do this. So we have to have some measure of when are the samples of our neural network, when are the output samples similar to the samples in our database. Now, in physics, we usually don't have a database, or at least we want to avoid having a database because we want to solve the sampling problem. So we instead have an energy function, and this energy function defines the probability density that we want to sample from. It's just difficult to do this. Well, so these are actually actual samples from such a generative model that's actually already a few years old. So these are much, much better now. So these are really made up samples of celebrities, and you can see, so these people don't exist, but they look important. So the network has learned something about celebrities. So this is using a method called normalizing flows. And this is very important for us because normalizing flows is a kind of neural network which is invertible. So that means we have inputs here, Z, that are drawn from some probability distribution that we can easily sample from. And then we can push these forward through the network and get samples X. So in X are our celebrities or our molecular configurations, so whatever we want to sample. And now this network is specific in the sense that we can also invert X and go back to Z. So it's a bijection. Yeah, and of course, you need a very specific neural network structure to be able to do this. But if you can do this, you can leverage the transformation of random variable rule for invertible transformations, which says that you can compute the probability of generating an X from the output, given the probability of generating a Z at the input, and the Jacobian of the transformation. So basically, the determinant of the matrix of the first derivatives. So if you can compute those efficiently, then you can use this, and then you can try to make the output distribution, because you can evaluate it pointwise. You can compute P of X. You can try to make that similar to our Boltzmann distribution. Yeah? And this is what Boltzmann generators do. So we can compute this from the network, and you can sample it from, you can sample X from the network, and you can compute P of X from the network. And you can then try to make this similar to our e to the minus u of x. Of course, we don't know the prefactor, but we'll try to get away with that without knowing the prefactor, usually. We can also reweight our network output to the Boltzmann distribution. Yeah? So we can use all kinds of statistical mechanics tricks that exist to reweight probability distributions to our target distributions, because we will never match these two exactly. Okay, so that's what a Boltzmann generator is. Use essentially a normalizing flow to generate output distributions, and then reweight those to the target distribution that we want to sample from, and try to get one-shot samples of our complicated target distribution this way. Yeah, just some ideas how we can use how we can build invertible neural networks. So normal neural networks are not invertible, of course, but what we can do is we can separate our variables in two groups and then transform one group as a function of the other group with an invertible transformation, such as multiplication or addition. So those are invertible by division or subtraction. And then we can write down the inputs as a function of the outputs by just inverting these green circles here. And we don't have to invert the blue boxes. And then we can swap these two channels around so that we nonlinearly transform everything as we go deeper, and then define deep networks in this manner. That's one approach. There are many, many approaches to do invertible neural network, but that's one simple approach. And so now what we can basically do is we can write down the Kullback-Leibler divergence, or also known as relative entropy, between the output distribution of the neural network and the Boltzmann distribution. And we minimize those. And there are essentially sort of two Kullback-Leibler distributions. So we can kind of swap the two distributions that we're comparing, and we get two different KL divergences. And one turns out to be identical to a maximum likelihood. And the other one is, we here call Kullback-Leibler divergence, or we also call energy-based training. So basically this one is perhaps the most interesting one, because it relies on taking samples at the input, and that's something we can easily do. Remember, sampling from a Gaussian distribution, maybe. We just push those through the neural network, which initially is, has random parameters, so it will initially just generate nonsense. And then we evaluate the energy of the samples we get at the output. And we essentially minimize the energy. But we don't only minimize the energy. If we would do that, we would just get essentially 1 point. We would collapse to the minimum energy. But we also have a part here which comes from the Jacobian of the neural network. And that turns out to be an entropy part. So we also maximize the entropy of the output distribution. So in other words, we're minimizing a free energy. And so this minimization of the free energy is satisfied when our neural network outputs exactly from the right distribution. In practice, we need to use a lot of tricks to avoid that this collapses to a few metastable states or one metastable state only. And this is why I have these different terms here. But that's the idea. And then we can do reweighting or importance sampling with the output. Okay, so some examples. So we can train with data from multiple metastable states, but we don't have to the right weighting between these metastable states. Yeah, so imagine you have just some samples from metastable state 1 and 2, but you have no idea what the free energy difference between them is. And also the transition between these states may be very complicated. You don't know a particular collective variable. It's not obvious which collective variables would be useful for doing some enhanced sampling between those states, but you still want to be able to compute the free energy difference. So maybe even the free energy profile in which these connect. So we can use them to train such a Boltzmann generator. This here is then the representation of points in the latent space, in the Z space. And then if we sample from our Gaussian distribution here, map back, reweight, then we can compute the unbiased free energy profile. We can also use this iteratively with some other sampling method. So this is not really for exploring new states, but we can combine that with other sampling methods that can find new states. And we can, for example, now formulate all our sampling models in these latent spaces. Now, the idea here is that these Boltzmann generators kind of transform our data. So they learn like a very general coordinate transform to a new space in which things are organized such that our metastable states, so the high probability long-lived states, are close together. Yeah, so the idea is this is a better representation of what we care about in this high-dimensional state space. Right? So now we can do anything that we like to do normally in X space in Z space instead. We could do replica exchange in Z space, or Metropolis Monte Carlo, or replica metadynamics, umbrella sampling, whatever. And because we always have this invertible map between latent space and real space, we can always rewrite any of these methods and any Metropolis acceptance step. Yeah. And so we can, for example, start with one point, start sampling around this point, learning the Boltzmann generator, starting to explore the space by sampling in latent space and retraining the Boltzmann generator, and then start to find new states this way. And yeah, so that's basically the idea here. Here's some more interesting examples. So this is a condensed matter system where there is a bistable dimer here, which is happy if the two beats are together or separated. So there's this one-dimensional energy for the separation of the two beats, but it's in a dense system, so there is not a simple Monte Carlo move to just open and close them, because you will always run into solvent particles if you try to do that in one step. But we can train a Boltzmann generator in the open and close states, and then sample both states in the free energy profile, connecting them actually at different temperatures. And yeah, can you go to more complicated systems? We hope so. So here is a protein in implicit solvent, BPTI. And this is the all-atom representation. And this is actually a sample from the Boltzmann generator. So this is really a one-shot sample where you generate a point in a 3000 dimensional Gaussian distribution, feed it through the neural network, and then you output this. And so this kind of looks okay for a chemist. You can also sample by different states that way. And here are some indicators that you get decent bond lengths and bond angles, etc. This is maybe still not at a stage where you can really use this for Monte Carlo sampling, because there's still not enough overlap between the energy distributions, but that is definitely getting there. So we hope we can use this to learn essentially good proposal steps for relatively large systems. By relatively large, I mean hundreds to maybe 1,000 atoms. So that is maybe not enough to simulate a protein, but we can maybe subdivide a protein into, or a solvent box into parts, and use some sort of Gibbs sampling where we use something like this to do essentially large scale moves on parts of the simulation system. So that's the idea here. Okay, so now I'm it's 10 minutes before 5, so it's up to you. I can briefly go over the last part, maybe in 5 minutes, just to
5 minutes. Fine. 5
Yeah?
minutes. Fine, Frank. Go ahead.
Okay, let's do that. So, because I mean, this is perhaps not like the mainstream topic for this audience, because I mean, I know many of you do quantum chemical calculations, but of course you're interested in biological systems, so you're interested in methods that can be really used in a QMMM setup. So, and this is here basically a method to really try to compute very accurate approximation of the electronic structure problem. But I think it's perhaps a good starting point to get very high accuracy benchmarks for machine learning methods that then can extrapolate using supervised learning to larger systems. And so this work has been led by Jan Hermann and Zeno Schätzle. And yeah, it's about solving the electronic structure problem, specifically the ground state electronic structure problem in the time-independent non-relativistic Schrödinger equation. So we have a Hamilton operator that defines our chemical system. And we want to solve the wave function and the energy. So we basically want to compute the eigenfunction and the eigenvalue. And now, of course, there is a whole set of quantum chemistry methods that do that, like DFT, coupled cluster, and many, many more. There is this field of learning from these methods, machine learning from these methods, where you basically use DFT or coupled cluster or some other methods to generate energies and perhaps forces or other quantities for a bunch of molecules, or 100,000 molecules or something like this. And then you use machine learning to train perhaps neural networks or kernel machines to predict those for new molecules that you haven't simulated yet. So these are basically machine learned force fields that are transferable within a certain class of chemistry at least. And this field is very advanced and can basically, if you stay, for example, within small organic molecules, you can reach the accuracy of the method that you use at the input to generate the ground truth data, such as DFT or coupled cluster, which also means you're limited by that. So whatever DFT gives you, which in many cases is not too bad, but can still be several kcals per mole off, even for a small molecule, and that could be traumatic if you want to compute thermodynamic properties such as binding or unbinding rates. Then you're limited by that. And so the question is, can we do something that is much more accurate here? And so turns out that you can actually use deep learning neural networks to solve this equation, or approximately solve this equation. And the hope is really to get a method here that is a bit of an outlier of the usual scaling law, where you get a little bit better by paying a lot more. This is a quantum Monte Carlo method, where you use a deep learning ansatz function for your wave function. And that's what we've developed here. So it uses, again, a variational approach to represent solutions of the electronic Schrödinger equation. So it says the energy of the ground state is the minimum over the expectation value of the energy over wave functions that we basically try out. And we represent these wave functions with a neural net. And this here becomes our loss function. And in Monte Carlo, we compute this expectation value by using Monte Carlo, by sampling from the electron density and then computing what's called the local energy, which is something we can compute easily if we have our Hamiltonian and if we have our wave function. And then we basically sample electron positions in real space to sample this expectation value, to generate our training data, and then we train by minimizing our variational energy. We train a neural network. And the main difficulty here is antisymmetry. Electrons are fermions, so the wave functions that solve that we should search over while minimizing the variational energy should be antisymmetric. So the sign of the wave function should change if we exchange two same spin electrons. And the usual approach to do this is slated determinants, such as in Hartree-Fock or coupled cluster or MP2. So we have basically our n electrons. We choose some orbitals, so some functions. We evaluate these orbitals in the electron positions, write the result in a Slater matrix, and compute its determinants. And this is called a Slater determinant. And this is by definition an antisymmetric function. Yeah, it changes sign if we change. Somebody's mic is on.
I muted that. Go ahead.
Okay, if it changes sign if we change the electron positions. But that basically means now if we fix the orbitals, we need eventually exponentially many Slater determinants, because there are exponentially many ways to do this assignment here to get really, really high accuracy. And instead, we use the so-called backflow. So we use a neural network which changes the positions of these electrons as a function of the positions of all other electrons. So we learn essentially a multi-body coordinate transformation of electrons and then enter the Slater determinants. And then we hope to be much more expressive and get away with much fewer Slater determinants. And that's the trick. So that's the Pauli net architecture. Not going to go through it in detail. Bottom line is we have neural network parts here that we can learn to represent the wave function. And we have kind of classical parts that really come from physics intuition and come from standard approaches to do quantum Monte Carlo, and that introduce certain things like asymptotic behavior, making sure that things are antisymmetric, making sure that we start from a good baseline solution, etc. So build physics in that essentially helps us to get good solutions without paying too much computing. And so the bottom line is we can get very high accuracy solutions of the electronic structure problem for small molecules with this approach, with orders of magnitude less later determinants than standard quantum Monte Carlo approaches. And so that, we hope, allows us to go into regimes of maybe a few hundred electrons where we can do small molecules with a super high accuracy, where we don't really have a black box quantum chemistry method that works kind of uniformly well. Most methods that work, that really squeeze out the last bits of the correlation energy, are highly tuned to the specific molecule. And essentially we want something that's more automatic and robust. Yep, and so with this, I want to wrap up. Just say a few words. Why the hell do we want to do deep learning? I've advertised this so much. So I think that the most important message, why do we do deep learning, is in all what we do as computational scientists are essentially computations, and usually with parameters, right? That's what we do since what this field does since decades. And deep learning, don't think about particular neural network architectures, so that's not so important. Deep learning is a generic way of taking any computation that has some sort of parameters associated with it and fitting them in an automatic way. Because we can represent any computation as a computational graph. And if we can differentiate through this graph, we can evaluate it forward and backward, and we can compute the error of some predictions with respect to some sort of ground truth or some sort of reference. And we can propagate it back to the parameters and adapt the parameters. So deep learning is an automated method of computation. It's basically the next level of what MATLAB and SciPy has done, MATLAB and NumPy have done for computation in general, or computational algebra packages in general. And that's why it's so powerful, because it's a really, really general approach to solving computational problems. Yeah, acknowledgements. Thanks a lot to my group members, to the funders and my collaborators. This is my group. I think this is the last group shot before COVID, and this is how we look since, and how everybody else looks since. So really hope that we can go back to that state at some point in the near future. And with that, thank you for your attention, and happy to take questions.
Thank you very much, Frank. We certainly hope that we can go back to to some normalcy soon, and then have you even here physically and can have some interaction and socialization. That's what we are missing most. Thank you very much for the great talk. Before actually I go to several questions that people have, let me actually take privilege here. So just, it was very interesting that you pointed out. I mean, usually people say these machine learning based approaches are limited by their training set and the domain that is used to train them. But it seems that now you introduce at least part of your talk that there are ways to at least somehow get out of that initial training set and sort of extrapolate this a little bit. The example of the celebrities that you used, that was very interesting and new to me. But I had one question. So like, I mean, I guess probably you are doing the best, most efficient sampling we can have in molecular simulation, you know, very long simulation, multiple simulation, Markov state modeling, and projecting them, clustering them, at the same time using machine learning. So what do you think is still missing in our business? Is it force field? Is it not having enough structure? What is the major missing piece in our business? What do you think after having so much sampling?
So I think sampling is still a big problem. I've not had any single project where sampling wasn't a bottleneck, and we're really trying to like not do very big systems, but a bit smaller systems where we can do very extensive sampling, and it's still always a bottleneck. I think one important step to get out of there is, so if you look at these really big systems that are interesting from a biological or biophysical perspective, like the ribosome, the spliceosome, all of these big machines, it's not useful, I think, to view these as from a Markov state model standpoint, where the global state of the machine is essentially one out of a list of states. Okay, why is that? Because I think when you are going to beyond a certain size, things start to uncouple or to weakly couple. Yeah, so even if we have like protein domains or entire proteins, and we describe each of them as a Markov state model, if you have enough proteins, again, you have kind of a combinatorial explosion of states. So you have to have a model at this higher level, which efficiently deals with them and is not trying to characterize everything by one state, because then that wouldn't be feasible. Then you would demand an infeasible amount of simulations to parameterize it. So we're starting to go in this direction also, and a few other people do. And so one way to think about this is, so there is this set of methods called Markov random fields or graphical models, where you basically have subsystems that each have like multiple states and that are coupled to each other. A simple example of that is an Ising model. In an Ising model, your individual spins have their own states, but their flip dynamics or the flip probability depends on the other spins it's coupled to. So I think that's an important way to think about these larger systems, so that we have some sort of, it's kind of like linear scaling tricks in quantum mechanics, like tricks to decompose the big systems into manageable parts, not only in time, but also in space. So that's an important.
Very nice perspective.
And of course, everything else you said, like force fields is still a problem. Like I think these are problems that are not going away anytime soon. So lots of NIH centers to run on those.
Sure. Thank you so much. Okay, Seper, do you want to ask your questions?
Sure. Thank you, Frank, for the great talk. So actually, I have two questions related to the Boltzmann generator. So for the protein that you show, I mean, I don't remember the name of the protein. For the protein you show, how do you construct the structure of the protein? Because to me, the loss function is the energy-based loss function. Mm-hmm. So how can you get the actual structure of the protein from Boltzmann generator?
Yeah, so in theory, the energy function is all you need. But of course, it's clear that if you would start sampling from completely nonsense structures, so essentially random noise, positioning the atoms randomly in space, you wouldn't be close enough to the actual distribution that you could converge to that. So here, it's very important to actually use a coordinate transformation. So remember, this is an invertible neural network. So we can do anything that preserves this invertibility. And one thing that preserves invertibility are, for example, things like instead of Cartesian coordinates, using internal coordinates, and using PCA. So here what we do is a combination of that. So we use PCA and whitening. So whitening means you basically do PCA and then you normalize all PCA directions to have unit variance. We do that for all the backbone coordinates, and that's an invertible transformation. And then we do an internal coordinate representation of the side chains. And that's also invertible, because you can equivalently represent the side chain coordinates by essentially by Z matrices, so bond lengths, angles, and torsions. And that's a very important step. And then we just train this neural network on top of that. And we also bootstrap the entire training with samples from the energy landscape. But the important thing here is we do not necessarily need to know the statistical weights between those samples. So we can just like use maybe different crystal structures or something like that to kind of bootstrap the method.
And another question that I had was related to the double well potential. So based on the maximum likelihood, loss function that you have, I mean, this is my understanding. The peak of the probabilities in the real space should be mapped to the peak of the Gaussian. I mean, this is what my understanding. I mean, you can tell me if I'm wrong or not. But if that's the case, why are there some yellow points in the actual space that you map them to latent space, and they are still close to the peak of the Gaussian?
So there are two different distributions here. One is the probability distribution in in the state space, where we have a Gaussian in the latent space and we have something non-Gaussian in the configuration space. Yeah, so if you would maximize probabilities there, then it's like you say, you find the maximum. But what we are maximizing is the likelihood for the parameters of the neural network. So it's basically the we are maximizing over the probability distributions that are possible. So we find the probability distribution that is closest to our target distribution. It's a distance between distributions.
Okay, thank you. Thanks.
Heon?
Hello, Professor Noe. Thank you for the talk. This is just to confirm my understanding. So the VAMPnet, when you're maximizing the autocorrelation, so are you actually trying to get like the tau there so that you can use it to, so I mean like,
no, you fix tau. Tau is a hyperparameter that you fix. That's the time scale at which you're building the model. You can build it for different tau.
So you fix the tau, and what you're just trying to get in the end is like f, the expression f.
Yeah. You're trying to find the mapping from your full state space to this low dimensional clustering space in which the Markov state model is built. So that's what the neural network represents. And then you also learn a matrix, the kind of the transition matrix, the MSM transition matrix. But you fix tau, and you can do this for multiple tau, and then it's like building a Markov state model for different lag times, and you can do the standard Markov state model analysis for different lag times, like look at time scales, etc.
You assume that there is a good tau, like before you actually apply the VAMPnet?
So you compute it for a range of taus, and a good tau means it's large enough so that you can get rid of a lot of the fast processes that you're not interested in, so that you can actually represent your problem with a few states. Basically, what tau does is if you remember this spectral decomposition of the dynamics, if you increase tau, then you have this exponentially decaying parts. So the larger you make tau, the more of these terms disappear from the sum. And that means the larger you make tau, the more and more fast relaxation processes disappear.
So less spectral clustering.
Spectral clustering to less clusters is possible. So the problem becomes lower dimensional in a way. It's still kind of a manifold in a high dimensional state space, but the manifold itself is lower dimensional. And that's important, because if it would be high dimensional, you couldn't do anything. So having some tau, some minimal tau is very important to make the problem feasible, also for Markov state models. But there's a limit, because if you push it too far, then essentially only the stationary process is left and you've lost all your dynamics. And then that may not be what you want, because probably you want to say something about transition rates and pathways and things like that. So there's a range of tau that's interesting.
Okay, thank you. One more. So for the Boltzmann generator, like when I'm trying to get that, the original space representation, and I choose a single data point in the latent space, the inversion, when I invert it from latent to original space, do I get like degenerate configurations, or is it like just unique. A specific?
It must be unique. Yeah, it must be one-to-one, because otherwise the transformation of random variables wouldn't hold if it wouldn't be a bijective function. There are ways to avoid doing that, to kind of start with lower dimensional and then go to higher dimensional spaces, and we've worked a little bit on that. So there's a paper called Stochastic Normalizing Flows, where you can do things like that using tricks from non-equilibrium StatMech. But in the basic setting, it has to be invertible and one-to-one.
Okay, thank you.
Dimon, maybe the last question.
Thank you for the talk. So I have two questions. So first one is, when I'm using the VAMPnets, so am I allowed to use like lesser amount of simulation data to get the equally accurate Markov state model, or is that possible?
Compared to what?
Compared to when I'm not using any neural network or any kind of a machine learning based optimizer to get the propagator. Suppose if I'm using some reaction coordinate of my own choice or something like that.
Not necessarily. I think the main benefit of using a VampNet over like a standard MSM is that you can automatically do the different steps of MSM construction, selecting features, dimension reduction, all of this stuff. You can do that automatically and in an optimal way. So it really helps you to find a high quality MSM. But the MSM itself is, I think, what helps you to use perhaps less simulation data or shorter simulations at least, compared to not using an MSM, because it allows you to compute the kinetics from conditional statistics. So what's the probability of being here given that we were there, and vice versa. Whereas if you don't use an MSM, and if you just do some sort of counting, let's say, or direct trajectory analysis, like doing a histogram or something like that, then you don't do that, and then you have to have single trajectories that are long enough to sample reversibly between your metastable states.
And I have another question on the Boltzmann generator. So how good are the barrier heights sampled in Boltzmann generator? Like if I want to get some kinetics of the transition between states, is that possible to get from Boltzmann generator?
It's possible, but it depends on how high these barriers are, because the aim of the Boltzmann generator is to get an equilibrium sample. Now, if the barriers are so high that they have negligible probability in equilibrium ensemble, you will not get samples out of the Boltzmann generator on the barrier. But so in those cases, we usually do not want an equilibrium sample. We want to bias our sample so that it's perhaps more uniformly distributed along a certain reaction coordinate. And you can do that too. So you can combine reaction coordinate loss with the Boltzmann generator if you're interested in a particular energy profile. And so that's also what we've done in that paper.
Mm-hmm.
Thank you.
Okay, Archie, the very last question.
This might be sort of a naive question, but I was just wondering how well this Boltzmann generator would work if you have like multiple metastable states, so over 10, maybe 20, 30, 50. Would there be any like problems or could be any issues that may arise? Or
so multiple metastable states is fine. It's just you need to keep in mind that the Boltzmann generator is not really an exploration method. So it will not by itself generate new metastable states it hasn't seen. So it's really a way to represent your data in a way that it can be efficiently sampled, so that you can efficiently compute free energy differences between different states when you initially didn't have this information. And it's best to couple it with a method that's good at exploring new states. So something like multi-temperature sampling, metadynamics, whatever.
Mm-hmm. Okay. Well, thank you very much again, Frank, for the very stimulating talk. And we have to stop at this point. It's already 15 minutes past. Good to see so many friends attracted to your talk.
Thank you very much. It was a pleasure.
Absolutely. Thank you again. Bye-bye.
Thank you, and see you all soon. Bye.